Hello everyone, thank you for joining us today. My name is Garding. I'm a field application scientist working for Covaris, and I cover epigenomics applications in Europe. And today, in this epigenetic exchange platform, I would like to give you an update on epigenomic workflows in Covaris. So the talk will be structured as followed. First, I will give you an update on the Covaris AFA protocol. So I will tell you how the AFA protocol can help you in standardizing your chip workflows and how you can pair it with other methods for most efficient sample preparation. And I will also let you know what we recently updated in our protocol to make it even better based on feedback from the field. Then I will talk about newest developments at Covaris and epigenomics, two projects which are still also in progress. One is partnering with another company called Arima, where I will talk about high-C and high-chip developments. And another project is a collaboration with the UCSD in the US, where we are working on speeding up chip workflows by accelerating IP to AFA. So let's start with the update on Covaris AFA protocol. So if we talk about the epigenetic and transcription factor landscape, we appreciate that this represents a key regulatory layer in the genome. And if we want to characterize this landscape, we can look at post-translation histone modifications, transcription factors, and pioneer transcription factors that bind to chromatin, or any epigenetic regulators such as readers, erasers, or modifiers, which can also modify chromatin structure. And during the recent years, CHIP has been proven a powerful tool to better characterize chromatin landscape. And if we look at the challenging steps of this protocol, we can briefly go through the workflow. So you can start with any sample input that you would like to know the chromatin structure. And this can be cell culture, tissue samples, primary cells. Then you fix it with formaldehyde in order to preserve the in vivo situation. Then you need from an efficient cell lysis to extract your nuclei, and then spoon chromatin shearing. And which is important to these workflows is that you need to control your cell density, the fixation step, the lysis and nuclear preparation, as well as the chromatin shearing process in order to have good starting material for your downstreamers. You perform shearing analysis and your chromatin immunoprecipitation, followed by a reverse cross-link DNA purification, and then your analysis, which can be qPCR or ChIP-seq. And Covaris helps you in the very important sample preparation step and gives you standardized solutions. So we have a true chip kit, which helps you all the way to get properly sheared and prepared chromatin. And this helps you to standardize your workflow. So the first step, as we already mentioned before, is that you cross-link your proteins to the DNA to preserve the in vivo situation. And in this cross-linking step, the Covaris AFA protocol provides you only monomeric formaldehyde because we are working with single usage formaldehyde. And this is important because as soon as formaldehyde comes into contact with oxygen, it will react and it will start forming oligomers and later polymers, and they are much less efficient in cross-linking. That means if you're starting out with a big bottle of formaldehyde, just by the formaldehyde reacting with the oxygen, you will get over time a less efficient cross-linking reagent, and this can impact on your signal-to-noise ratio in your ChIP-seq experiment. This is why we strongly recommend to work with single-usage formaldehyde for chip assays. Then you perform your chromatin preparation. For this, the chip kit will provide you optimized buffers for a very efficient and reproducible chromatin preparation. And then you will shear your chromatin. And if you shear the chromatin with the Covaris device, you will have a non-contact shearing, random shearing, and isothermal shearing. Isothermal is important because you will not damage any epitope due to heating, and more importantly, in chip assay, you would not reverse crosslinks that you introduce to preserve the in vivo situation. If you would be heating your sample during the shearing process, you might reverse some crosslinks, and then you will later on not see all the binding sites of your factor, and for scarce factors, you might be losing binding completely. There is a focused supply of energy. You have a highly reproducible shearing pattern, which is important if you want to compare sample to sample, lab to lab, and experiment to experiment. And again, you have the unique epitope preservation, which mainly results from the isothermal processing. After this, you need to pull down your protein DNA complex. And here, the Covaris kit has a universal pairing with any commercial or homebrew method. So you can use our sample preparation independent of the workflow you want to use downstream. And one advantage of the kit is that you need to dilute your chromatin less than with any other method, because we are using only 0.1% SDS in our shearing buffer. And therefore, you only need to dilute your chromatin 1:1 for later applications. And this gives you a better chromatin antibody ratio, and therefore leads also to better IP efficiency, especially when you're working with limited sample input. Do you reverse your crosslink, do your DNA purification, and your analysis of interest such as PCR and sequencing? So I told you that this protocol helps you in chromatin sample preparation and shearing, but due to feedback from the field, we implemented a few changes to the kit, and I would like to briefly go about these changes. So if you in our days go to our web page, you will find for the TrueChip protocol that you would find a protocol if you purchased the kit prior to November 2019 or after November 2019. And this one will give you the latest protocol. And in the protocol on the first page, you will see all changes that we have implemented, but I would briefly go about them right now, and they are rather simple. So first of all, we are now providing our buffer F, what is the protease inhibitor cocktail, as a powder and not anymore as a dissolved reagent. And this allows us for a shipping at room temperature and a better stability. Meaning that if your kit is for any reason stuck somewhere, let's say at customs, you wouldn't have any issue that it has to be stored cold. So upon arrival, you will dissolve the powder yourself with water, and we outline exactly which water you should be using in the kit. And then it will be stable at 4 degrees for 1 to 2 weeks, or at -20 for 12 weeks. And the only thing that you have to keep in mind if you are used to work with our old kit is that now you would be dissolving the powder according to a 50x stock, and beforehand we provided a 100x stock. So if you are switching now, you should just use double the amount of protease inhibitor cocktail. And since this is only very little amount, like microliters, you would not dilute your chromatin much more, so it's not impacting your results. Furthermore, we have included a new buffer, which is called a buffer G, and this is a reverse cross-linking buffer. First of all, if you are following normal chip-seq experiments and you are not doing any shearing optimization in buffer DT, you would not need this buffer at all. This buffer is only meant if you are starting up with a new cell line and you are doing a shearing time course to establish your optimal settings. Then you would do your reverse crosslink in buffer D3. And what we found is that this buffer is not optimal for storage overnight at 65 degrees because the pH and the salt is not buffered optimally because this buffer was optimized for proper shearing. So this could cause some degradation, and you might be seeing that in your non-sheared sample. But if you now add the buffer D to your sample in the D3 buffer when you perform the reverse cross-linking, this will be perfectly preserved. So this is just meant if you are doing your shearing samples. And furthermore, we have now adjusted a few settings and experimental conditions based on more work that we have done. So we have added now settings for our 96-well instruments, for the LE220 and the LE220 Plus in the manual. We also urge you, if you're working with our lower power instruments for chips, so the M series, that you should not exceed 15 million cells in the 1 ml tubes for reproducible results. And also, we mention in a note that for transcription factors, you might have to adjust your fixation time because you might have very sparse factors with a short on-time on chromatin, or factors that are just binding transiently to chromatin. And then you might have to adjust your fixation time as 5 or 10 minutes might not be enough for certain factors. The next thing that we did is that we already implemented the 2x dilution buffer into the kit recently. And this buffer is needed because the buffer D3, in which we perform chromatin shearing, still contains 0.1% SDS, which needs to be sequestered with tritone. And as you can see, this buffer doesn't have physiological salt concentration, which is required for the antibody for proper IP. And therefore, you should adjust your sodium chloride concentration to 150 millimolar, to a physiological salt concentration, and you should add tritone in order to sequester the remaining SDS. And we have added this buffer, but we thought in order to make this easier for you to follow, to also put a schematic here. So you're starting off with your sheared chromatin in D3 buffer, and then you can either go with the 2x dilution buffer, dilute 101, and then go into your IP of interest. Or if you have very sparse samples and you don't want to highly dilute them, even though 1:1 might not be high, you can still prepare your own dilution buffer, which could be higher concentrated, but it's important that you would end up in the final concentration of sodium chloride of 150 millimolar and a tritone concentration of 1%. And the last thing that we implemented into the kit is that for most of our instruments, we have now ready to go settings for the Nexon protocol, which many people adopt in order to make their sample preparation better, especially when starting from difficult cells. But I will go over the method just in my next slides to bring everyone on the same page. What you can see is that for our M instruments and the SNE series, we have already settings because we have thoroughly tested them. If you are working with the LE and the LE220 Plus, please reach out to our application team or to me directly, and we can already give you settings that are working for most people, but they might need a bit more of an adjustment as we haven't tested them rigorously in the field. So, what is the Nexon method, and when does it help in sample preparation? So, I already explained to you our chip kit, which really helps in standardizing sample preparation workflows. However, this normal nuclear extraction with only chemical buffers could be inefficient for some cell types. So we know there is some hard-to-lyse cell types around, such as PBMCs, primary cells, or very mesenchymal cells, which could be resistant to chemical lysis only, and then you would end up with an inefficient chromatin preparation, and sometimes you might also remain a non-sheared chromatin peak. And this is because these cell types are very hard to lyse, and you would, during your procedure, not end up in an efficient nuclear preparation. And therefore, the MPI in Freiburg has developed the Nexon protocol. Nexon means nuclear extraction by sonication. And you can follow this paper for more details. But what they literally do is that they start over with fixed cells, and then during their nuclear extraction, they put the chemical lysis buffer, but they support the chemical lysis with mechanical lysis by supplying Covaris AFA. So they put the reaction into a Covaris tube and put it in our ultrasonicators and apply AFA. And for most cell types, this needs around 60 to 90 seconds. You will appreciate that in the paper. However, for example, for T cells, you require around 5 minutes of AFA to get 80% of cells nuclei extracted. And these are cell types where a lot of people struggle with chemical lysis only. And this is mostly because chemical lysis usually works in a way that you make holes into the cellular membrane, and then you use hypotonic buffers in order to burst the damaged cell membrane. However, if you have cells with a very, very small rim of cytosols, such as PBMCs, there is also not enough power of the hypotonic effect to really burst the cells efficiently. And therefore, it makes a lot of sense to support this process mechanically by AFA. And this then leads to very efficient lysis of the cells. Less sample input is required, and you can standardize your epigenomic applications. What you should do when doing this with a new cell type that you are not used to, after every minute, you should take an aliquot, like 10 microliters of the reaction, and watch it with DAPI under the microscope just to see how many nuclei did you extract. And in this paper, you can see a very nice description of this, very nice pictures, and you can follow for a lot of different cell types how this would be looking if you have non-lyse cells and lyse cells. So there's already lots of publications around who have been adopting this method for chip. However, this method is not only useful if you want to do chip, it's useful for any method where you need to extract efficiently nuclei. So there's more and more publications also showing this for in situ Hi-C. And you can think of any of your own workflows that you have in mind where you need to extract efficiently nuclei that this could be applied. But for a chip workflow, you would now use the Covaris AFA in 2 steps of the workflow. First of all, to isolate the nuclei, and then for chromatin shearing. So we have lots of customers been very happy with this method, and we have been working with Bayer in order to standardize their chip workflows internally. However, what we figured out there is that some cell types, like hepatocytes or adipocytes, they have a lot of storage of, for example, lipids. And this makes these cells difficult to work with if you are thinking of chromatin shearing, because these lipids make everything very viscous. And since they are not the same in every sample type, they could make shearing non-reproducible. And therefore, we have escalated this workflow a bit further. So we isolate nuclei with AFA, so like in the next one, everything in the true chip kit. Then we perform a sucrose centrifugation in order to purify the nuclei, and then we perform chromatin shearing again with AFA. Then the results have been very good. If you have cell types that are still very difficult to handle, reach out to us, and I can provide you this application note that we did with Bayer, which describes every buffer and everything that has been used. So this has been their sequencing data, so they are so far very happy with the workflow. So I would like to briefly let you know that which kind of sample inputs does Covaris support in chip. which solutions do we have? So if you start over with mammalian samples, we have the TrueChip kit that I already explained. You can work with 100,000 to 30 million cells. And we have a protocol for ultra-low input, so whenever you start working with less than 100,000 cells. We furthermore have a solution for fresh frozen tissue ranging from 20 to 120 milligrams. And we also have a solution if you want to perform chromatin immunoclassification for FFPE samples of formalin-fixed paraffin embedded samples. And we have also more specialized chip applications where I can provide you internal literature and testing. For native chip, we also have a kit around. And if you want to work with protein complexes, I can provide you an application note for double cross-linking. We have lots of literature about the next one, and I also presented to you the application of this layer, which involves another sucrose purification step for the nuclei. If you want to start over with non-mammalian cells, we have solutions for every organism. For Drosophila, we have a very nice application note if you also want to start with embryos and very low input. We have application notes for yeast and C. elegans, and for all other cell types, and also these applications, there's lots of publications where we can provide you the literature, which mostly provides direct settings that you can use for certain cell types, where you can start with your cell type, or you can directly reach out to the authors of the publication. And the nice part about Covaris devices is that if you find settings in a publication, you can use them on your device if you have the same instrument. And if you have a different instrument, just reach out to our applications team and we can calculate your settings, which you can use on your device. So I mentioned that we have solutions in starting from fresh frozen material. And I would like to go about that a bit more in detail. So how do we support epigenomics applications from fresh frozen tissue? So if you start with a frozen tissue, we have another instrument which is not an ultrasonicator. It's the so-called cryoprep, which enables reproducible chip from in vivo primary material. So it's a tissue pulverization system. So first of all, we provide you with specific bags which are out of the material where you can put your sample inside. Then this material can resist very low temperature, so you can dip it into liquid nitrogen. And even when stored in liquid nitrogen, you can transfer, it is resistant to very strong mechanical forces. So you can then put your sample into this electric hammer where you can choose 6 different forces with which your sample is impacted. Put your sample inside, so apply the impact, and then your sample is pulverized in this bag, meaning that during the entire process, this is a contact-free processing, so you would not have to do any grinding, you would not have to use water pistol where everything comes in contact, and obviously you wouldn't have any heating because this process of impacting takes only milliseconds. Then you have fine powder in your pouch, and you can transfer it to the transfer tube, and then you have your powder with which you can work further. So now you can either distribute the powder for different downstream applications, and it's quite nice because it's not in any buffer yet. So you can use part of the tissue for NGS, part of the tissue for expression analysis, part of the tissue for DNA methylation analysis, and part of the tissue for chip analysis. So if you want to proceed further with chip, we have a specific kit for that. It's the tissue TrueChip kit. The buffer composition is optimized for processing tissue. And what you need to know also is that the shearing buffer contains a bit higher SDS. That contains 0.25% SDS. so you would end up with a 1:3 dilution before IP. And then you can perform your chromatin shearing on a Covaris ultrasonication device. And as I mentioned, the possibility that you can distribute the powder allows for nice multi-omics approaches, and we have several people using this opportunity, and I would like to go through one example. So this is from a group who uses our E220 shearing device and the cryoprep, as well as the two-chip tissue kit that I explained beforehand. So what they are doing, they are working on mouse hippocampus. So they extract the hippocampal regions, and then they pulverize the tissue in the cryoprep, and then they distribute part of the tissue for chromatin analysis, so they did in this paper NPAS1 and NPAS3 and some part of the tissue for RNA analysis. And what we hear from scientists, what is nice about it is that you don't need to cut certain subparts of the tissue for one and the other analysis, but you have a powder which is actually representing all the different subregions of the tissue, and then you distribute that powder. So this gives you a very nice correlation of the data. Yeah, and then they performed their integrative analysis after ChIP-seq and RNA-seq analysis from this powder. So now I think one question for you could be like, where can we find the collaborative work that is done with Covaris? And you can find it on our webpage. So if you go to the Covaris webpage, go to resources, then go to technical literature, and then you can find 3D chromatin organization and some other epigenomics workflows. And you can find all the chip workflows where we have done collaborative work with scientists in the field. You can, for example, find the workflow that we did with Bayer, or also our chip workflow where you can start over with FFPE samples. So, and we are currently also working on an epigenomics brochure, which will be released soon. And you can reach out to us to get that. And for the participants of this webinar, I will send you the link as soon as the epigenomics brochure is ready. So, in summary for this first part of my talk, I have shown you that Covaris TrueChip allows standardized sample preparation. We have recently included a few optimizations. So the protease inhibitor cocktail is now provided as a powder. For people who are doing chromatin shearing analysis time course in D3 buffer, we now provide a reverse cross-linking buffer. We provide some optimized settings, including settings for the LE220+. We have provided a schematic for IP dilution. We provide natural settings for all instruments except the LE220+. And the next one itself provides a solution for more difficult to lyse cells where you can support chemical lysis as mechanical lysis. And I have shown you the cryoprep tissue pulverization system, which improves chip from tissues and also allows for multi-omic approaches. And I told you that the epigenomics brochure is coming soon. So now I'd like to take the remaining time to tell you more about newest developments at Covaris, ongoing projects. And one of them is centered around high chip workflows. So if we think about deciphering the 3D genome, you would look at the 3D organization, and here you can work with high-field capture in order to see which regions do come into close contact due to folding, which are not in close contact in the linear space. And you can look with methods like high-chip or ChIP for the proteins which are involved in these contact points. And as you can appreciate, lots of these methods involve shearing processes using the Covaris devices. And why people are choosing Covaris for these applications is because it's a tunable instrument, so you can tune the energy not only in one or two steps, but you can completely adapt the energy to the process that you need to do. It's random, it's reproducible, and it gives you a very tight size distribution, meaning that for these kind of assays, you don't need to do an additional size selection step in the end. So you can directly work with the Covaris shear material. And overall, you can achieve shorter shearing times and better epitope preservation, what is again very important if you want to do, for example, high chip. So how does the high chip workflow look? So you start over again with your fixed cells, and then you perform the nuclear isolation. And as I mentioned before, many people use the Nexon protocol for that. And then you would do the in situ Hi-C, meaning that you end up with your ligated and biotinylated contacts. And you use Covaris AFA in order to lyse the nuclei and perform a shearing. And then you perform your chip assay, the reverse cross-linking DNA purification, and the pull-down of the biotinylated fragments. And the advantages of AFA in this workflow are that you would have good epitope preservation due to isothermal processing. You would have very tight size distribution in the shearing, not requiring additional size selection. You would have very reproducible shearing. And overall, since you are retrieving all your material intact with such a good epitope preservation, it is ideal when starting with small input amounts. So where does the Covaris TrueChip kit fit into the high chip workflow? So in order to have an entirely standardized workflow, we have teamed up with ARIMA, and so for the entire workflow that I just showed you before, this workflow uses now the Covaris device for the shearing process that I outlined before. And that provides, this combination of ARIMA and Covaris provides you with a standardized, easy, and fast high-chip workflow. And we can pair both of our expertise in this workflow. So now we want to show you some 3D maps to show you that indeed this workflow uses very nice and reproducible 3D maps. So this is now H3K27 acetylation and H3K4 Me3 high chip, and we are looking at the MIC locus. And as you can see, if we look in terms, so we see very nice and reproducible patterns already by looking at the patterns itself. If we look into the bioinformatics analysis, we see for both marks in two replicates that the reproducibility rate is around 80%. So that means loops that are found reproducibly in both replicates. So now how many reads are required for reproducible loop detection? So if we compare to high-C assays, the high chip can retrieve maybe the more, let's say, functional interactions. So overall, you are enriching for informative regions. So for K27 acetylation, which would cover enhancers as well as promoters, you require around 300 million reads, and for the promoter mark, K4Me3, you would need around 100 million reads. And this has all been done in lymphoblasts. And if we compare this among different sample types, you see that this is rather stable, independent of the sample type. And the next question was that can we actually see cell type specific differences? And we compared lymphoblasts and excitatory neurons, And what you see, just looking at the ITGB1, BT1 locus, that you will indeed see the specific loops that you are expecting for excitatory neurons, as well as specific loops that you are more expecting for lymphoblasts. So to sum up this first part on the high chip development, so the pairing of a reamer and the true-to workflow allows for a standardized, fast, and easy high chip assay. We retrieve highly reproducible data and can reveal cell type-specific differences. Furthermore, we show that this is a cost-efficient solution that can help you. So currently, this assay is starting with 3 million cells. And you've seen we have been looking in histone modifications. However, we are starting to optimize further to work with less cells or less abundant transcription factors. So these are the works that are still in progress. So the second part of my talk is that I would like to tell you how we are speeding up chips by accelerating the IP to AFA. So what does that mean? So I told you in the very beginning of my talk that Covaris helps you in getting a standardized sample preparation step. So to give you the best starting material for your downstream analysis. However, due to some internal projects that we have, we knew that we can also enhance Brownian motion using AFA, because I told you before that the AFA system is not only high power. You can tune the energy that you're putting into your sample as you like. So if you provide only very soft AFA in your sample, you are just actually introducing some kind of mixing in your sample very softly. And this mixing, very soft mixing of molecules can enhance Brownian motion and thereby bring molecules in closer contact to each other, which would otherwise by the diffusion need much longer to reach each other. So knowing this, we were thinking whether we can accelerate the IT step in the chip, moving it from a 3-day protocol to a 1.5-day protocol. And we teamed up with a team from Alan Goran at the UCSD to conduct this project. So what we have done is we have shortened the incubation step with the antibody to an hour using soft AFA and compared to overnight incubation and encode data from the same starting. So as we have performed this for K4-ME3, K27-ME3, K9 acetylation, K27 acetylation, and CPCF. And what you can appreciate from these browser tracks is that the patterns look very, very similar to each other. However, anyone can show a nice browser track. So how does the genome-wide enrichment compare? So if we look into these plots, we see that we would always have the overnight incubation on the y-axis and the 1-hour AFA on the x-axis. And for every mark, we see that there is actually quite nice correlation, meaning that the data are very similar to each other. So the only mark that doesn't look very good yet, and we are working on that, is the K27 acetylation mark. So we see that the data don't correlate 100%, meaning that we might have to prolong the AFA treatment to maybe 1.5, 2 hours and see whether this is looking better. But from the browser text, remember, we still see nice enrichment at when we look into the real peaks. So now, if we look into the enrichment plots, we see again that the enrichment plots also look very comparable, and you might, so again, for K27 acetylation, we see less enrichment overall, but the same pattern. So we really believe that it is just a matter of adjusting the time here. However, for K27 MEG mark and K9 acetylation larger and CTCF, we actually see more enrichment at the binding site. So we are thinking that we might even improving the data here because we are enhancing the Brownian motion. So overall, this has been working very nice, and we are very confident that for most marks we can reduce the time to an hour, and for some others we might just have to use 2 hours or 3 hours, which is still much shorter than overnight incubation. So you're thinking about this workflow, you might think, oh, in one sample, this might maybe not really help me because for IT I would usually have more samples. So I would just like to update you again on the Covaris devices and which throughput do we support. So we have the single sample devices. This is the M220 for low power and the S220 for high power. Then we have the big brother of the M220, which is the ME220, which can handle 8 samples, and the E220 evolution, which is the big brother of the S220, which can also handle 8 samples. And then we have also sample ultrasonic that can handle up to 96 samples at the time. And for these 96 sample instruments, you can, for chromatin shearing, process up to 24 samples in 1 ml, which can work with up to 30 million cells. If you work with less cells, let's say up to 3 million cells, you can already move into the 8-strip format, so 130 microliter volume or 96 format. So this is 96 times 130 microliters, so you can work with up to 3 million cells in this per reaction. And this is obviously also the good solution for the mixing steps. So, and here you can just see some publications with booster systems. So, to sum up the third part of my talk, the IP step can be shortened from overnight to an hour. The K27 MEG, CTCF, and K9 acetylation display even better enrichment plots. For K27 acetylation, we need to optimize the timing, and chromatin shearing and IP can be performed in 96 format. So now I'm at the end of my talk, and I would like to thank you for your participation. And if you have any questions, please feel free to reach out to us anytime, either to me directly or to my colleague Rebecca. And this is like if you have any questions regarding your customer service. Thank you very much for listening, and I'm happy to plan further projects with you. Thank you.